Okay, welcome to Neurocare Academy. My name is Dr. Trevor Brown, and I have the pleasure of having with me Stefanie Enriquez-Geppert, who's a doctor at an assistant professor at the Department of Clinical Neuropsychology, University of Groningen in the Netherlands. Welcome, Stefanie. Thanks for joining us.
Yeah, hello. Thanks for having me here today. So many greetings from the Netherlands and from Groningen. I'm very happy to join today the interview.
Fantastic. Now, it's wonderful to have you. I know a lot of your work, and I think people listening to this are going to learn a lot about particularly neurofeedback, but also BCI and all sorts of different executive function research that you've done, so working memory, etc. And I'm really excited to hear what you have to say. So thanks for joining us. But before we get stuck into all that research, tell us a little bit about yourself. Where did you complete your university education? Where did you grow up? That sort of thing.
Mm-hmm. These are several questions. I prepared it directly for where I studied, so I think that is very interesting. Then I tell you the rest. So I like to answer this question a lot because I studied and did my PhD in Münster. So Münster is a very, very beautiful city in North Rhine-Westphalia. I don't know if you have ever been there. So it's in Germany, and it's a small city compared to many of other cities, 300 inhabitants. And what is really interesting about it is that there are three times more bicycles than inhabitants. And this is a little bit, you will see that I have mainly lived in bicycle cities, and this is kind of the start. And I brought you also a picture. I think that might be interesting. So maybe you want to go there, for holidays as well. So the university itself has more than 43,000 students. So this is also a little bit interesting as background. And you see there on the upper slides, you see a little bit an impression how Münster look like. So I can really recommend it for a day trip. So it's very medieval. You see then also the Institute of Psychology, and then I thought I brought you a little bit of what I did. So I completed my bachelor and my master in psychology with a specialization on clinical psychology and cognitive neuroscience. And here you see some pictures that we took. So everyone that you see there are the researchers. So this is research material that we use. And it was a study with MEG, so with magnetencephalogram, on statistical associative learning. It's a word learning procedure that you can also use as a therapeutic language approach for aphasia. And here you see also a map. So if you want to look up where Münster is, it's there, this red dot, and I really can recommend it.
Fantastic. Thank you. Yeah, I've spent a lot of time in Europe myself. I haven't been to Münster, but spent a little bit of time in Munich, and lovely part of the world. So lovely old towns, old cities there. So have you been to Melbourne?
To where?
To Melbourne, Australia.
Yes, of course, I have been. A couple of years ago, I really liked it. It was a little bit cold at that time, and it was a conference, the Human Brain Conference, Human Brain Mapping Conference. Very beautiful, yes.
Fantastic. Okay, so look, you've already started to talk a little bit about your research there in Münster. You went on to do your PhD after the masters there, and have you always wanted to be involved in research, or did you think clinically about the application? What were your thoughts there?
Yeah, to be honest, I think yes, it was from the beginning on one idea, and the reason for it was that we had a really super interesting course where we were invited to rethink how to improve existing research articles. And there was by coincidence an article about about language, how we acquire language, how we represent words. And this a missing piece was that you would have to study maybe languages where the syntax, so the grammar, is totally different. And by coincidence, I figured out that there is population in Mexico speaking Yucatec, where also the grammar is very different. So what I did is that I said, I want to go and I want to make this field trip, and I want to also assess this population. And that was really like an adventure. And from that beginning on, from that moment on, I thought, okay, research is really adventure. It's discovering, it's exploration, and I really totally fall in love with research. But I think also with a lot of role models around me, a lot of interesting researchers. Yes, but from very, very early on from the studies, I thought, yeah, this is really my dream, and this is, where I would like to go and where I would like to work.
Fantastic. And so that led you to take up research and start your PhD. Would you like to tell us a little bit about that now?
Well, I continued in Münster doing my PhD. And here you see one slide and this and one picture of the Institute for Biomagnetism and Biosignal Analysis. So I guess there are two things that are a little bit distracting. On the one hand, you might think, okay, this is a kind of a Swedish sauna. But no, this is a research institute, and it was on purpose built like that because it hosts the MEG. And sometimes people got also a little bit distracted by the name. So once in the office, we indeed got a phone call by a lady who wanted to buy a big magnet. But what the research institute is about, it is mainly focusing on translational cognitive neuroscience. So MEG, EEG, MRI, and TMS are the research methods. And I did my PhD on the subject of cognitive neuroscience. So you see it here on the right, and we're going to tell you also a little bit more about it. I shifted a little bit the topic. So what you see is that it's about cognitive control, it's about executive functions. So I investigated two of them, inhibition and conflict monitoring with EEG, structural and functional MRI. And so this is basically, what I did during the 3 times there, still in Münster, but at another institute. Okay, and what you see here are 2 articles that we wrote at the time in Münster. The first one is an article about conflicts and inhibition that differently affect the N200 P300 complex. And I think some of you know that the N200 P300 complex reflects one of the most important ERP complexes, and also, for instance, an inhibition paradigms. But the big question there was, okay, what does it mean? What is the functional meaning of the N200 and P300? And typically, this is something regarded as an index of inhibition. And this has definitely also an implication for clinical research, because if we would like to assess patients with this paradigm, we would like to know, is this maybe an index that shows us what is going wrong with this patient? So which functions are affected and that lead to difficulties maybe also in real life. And so here we selected a very nice approach, and this is an approach that combines MEG and basically MRI and EEG. And normally you say, okay, MRI has a very nice resolution in space, whereas EEG has a very good resolution in time. So what we did is that we combined it, and here you see on the upper right corner, you see our approach, what we did. So what we did is that for each participant, we had a realistic head model. We also assessed where exactly the positions of the EEG electrodes are. And this was definitely something where we learned a lot, or I learned a lot, because it's combining 2 different methods and then getting out really nice results. And we could then see, okay, the N200 seems to be something totally different than the P300. And I think, I would say that, this showed us really a lot, and this combines the strength of 2 different methodologies. So one of our colleagues at the Institute for Biomagnetism and Biosignal Analysis, so this is Professor Carsten Wolters, he has, for instance, a background in mathematics, and he uses also very similar, highly sophisticated individual models, but even that for pre-surgery, and for epileptic diagnosis, so before surgery. So this is one article where I think we learned a lot also about how to use methods in neuroscience, how to take advantages of different methods and combine them. And then I think another article where we learned a lot is the second one, variations in the mid-cingulate morphology. And for those of you who do not know where the mid-cingulate cortex is, I thought I'd show you a little bit one of the brain models that we have so that you have an idea. All right, so here you see a sagittal view of the brain, and the cingulum is kind of the belt around the corpus callosum. We're taking a look into this region. And what is interesting is that if you would take a look into MRI pictures, images of patients, you see that there's a big variation. So this part, the part of the anatomy will be different. So Trevor, your MCC is different than probably mine, and there are even more differences with patients. So the big question is, of course, what does it mean? Okay, and so here we have another graphic where you can see that there are differences also in gross morphology. So sometimes there can be even a doubling of a gyri, sometimes not. And we compared people that had different gross morphology. And then this way we manipulated basically the brain regions and compared the effects of anatomy on neurophysiology and then lastly on behavior. And this is also a foundation. Again, it has clinical implications because you will find that there will be differences in the mid-cingulate cortex across individuals, but also in groups of patients. And it can contribute to understand the psychopathology, but also help when it comes to diagnostics or therapies. So therefore, I think these are two studies that helped me a lot to understand what the neural underpinnings are of cognition, how they could contribute also to psychopathology.
Fantastic. Yeah, look, I think this sort of assessment, it's a great combination of lots of different techniques. You've got the temporal resolution of EEG, the spatial resolution of MRI. The clinical application of this, do you think it's possible in the future to be able to use this sort of assessment to predict outcomes of interventions at all? If you've got different size MCCs and the different effects that it has on the P300 or the N200, do you think that can help us? Okay, so we'll start now, and you can answer that question, Stefanie.
Mm-hmm. Yeah, it definitely can help us to understand several aspects. Let's say, if we look, for instance, at neurofeedback, what we see is that not everyone learns at the same pace. And there are even people that are very slow and people that even don't respond to the training. So they are sitting there 1, 2, 3 sessions. They try to modulate the brain activity, but it does not work. So we started to wonder what the reason is. And there maybe, there will be many different factors that contribute to it. So psychological factors, health-related factors maybe. I think one aspect could also be just the region, so just the generator, the area that produces this brain activity. So I would like to show you one graphic. So what you see here is that we indeed investigated this. So we knew the mid-cingulate cortex is very crucial for cognitive control, for executive functions, and it is also the generator of theta. Theta oscillations are kind of the working language of executive functions in a big network that implements executive functions. So what you see here is that we assess the volume of the single individuals participating in a neurofeedback experiment. And indeed, the volume of the right mid-cingulate cortex, it reflected how well, or it was associated on how well people would respond and would learn in a neurofeedback protocol how to increase their frontal midline theta. So there is indeed a relation also of anatomy on how well you can self-regulate a specific brain activity.
Would you like to keep going there, Stefanie? Well, I'm a bit muddled with my slides now, but are you going to talk about some of your previous research now, the slide with the rat. And
Yeah.
yeah. Okay. How would you like me to lead on to that?
Well, basically it belongs a little bit to the research achievements. I can continue with it and tell what I consider as an important next study. So we can do that. Or you can also re-ask. That's fine for me.
Yep. Okay. I'll re-ask and you can reshare. No problem. So, fantastic. So there is a clinical application of that, and hopefully it can help us to really personalize intervention in the future by not wasting time, I guess, doing interventions that we think are less likely to work. I mean, obviously it doesn't mean it won't work, but obviously there may be certain treatments that have a higher likelihood of actually being, a positive intervention for that individual. So all of these sort of biomarkers for prognostics are really key for us. So that's fantastic. So what about your other achievements up until this point, Stefanie? Would you like to tell us about some other research you've done previously?
Yeah, so basically, the study that I just mentioned was part of a series of research on neurofeedback, and for this, I moved to another lab, and it's an oscillation lab. So I had another new perspective on it. Well, and in this lab, this is a lab in Oldenburg, another bicycle city in Germany. And in this lab, everything had to oscillate. So my former boss at that time said to me, Stefanie, you're free to do whatever you want, but it has to oscillate. So I thought, okay, nice. I will gonna take a look on executive functions. And I would like to see what are oscillations that are important for executive functions. And with oscillations, in contrast to event-related potential, this gives us a big, it's kind of a treasure because there's a lot of animal research about it. And with animal research, we have a much better view in which brain regions might contribute to the implementation of an easy cognitive task. And what you see here is that I would like to show you one model. And this is a model about task switching. So it represents a little bit, cognitive, let's say cognitive flexibility in the human. And what you can see here is that there are 3 regions. So it does not matter too much in the moment what regions there are. But the dots and the sides of the dots show activity in the theta domain. And the theta domain or the theta increases if there is information transmission from one brain region to the other. And here at the bottom, I showed you, I have also a model of how conflict monitoring. So this is exactly, again, one specific function for executive functions or cognitive control might work. So this is a model of the neuro implementation. Then I have here kind of one graphic representing that when there is executive functioning or cognitive control needed, then you can see that there is an activity that you can measure in the domain of frontal midline theta. So it pops up, it is very short, but it reflects that, the brain detects that there might be a conflict. And then a lot of different processes are triggered. So with this, we thought, or we recognized that theta is important for executive functions. And with this, we set up then a research line on neurofeedback. So what we learned first is that we have a nice feature, brain feature that is associated to executive function and to cognitive control. And each time the behavior, the performance is increased and is better, then you can see also that there is an increase of frontal midline theta. So that means we have on the one hand, one brain feature that is a working language that is important, that each time when it is increased, there is a better performance. So this gives us an optimal target for neurofeedback. All right, so with frontal midline theta, so we have there a nice construct validity of the feature. We know that it is associated to executive functions. So, but the big question is, okay, if we just identified one important brain feature, can we also train it, for instance, with neurofeedback or not? So is it something where you really can get self-control over or not? And so what we did is we invited a couple of participants to come for 8 sessions. And we had a group that received feedback about their about their brain activity in a pseudo neurofeedback group that instead of a real feedback, they got a replay of the experimental group. And here on the left side, you see the graphic, and that shows that over the course of the training, there is a kind of increase in both groups, you might also say. But the neurofeedback group shows a much higher increase. So that means that giving real feedback enables participants to get self-control about this brain feature. And this is very nice for us researchers, but I think for everyone out there in the clinical domain, you might ask, okay, does it also have an effect? Does it transfer to cognition at all? And we assessed 4 different functions. And what you can see here on the right side is that it did not transfer to all of the functions. And this is also interesting to assess, why not to all of them? But it transferred to those functions where you need more proactive control. So it was the 3-bag task and the letter number. So there we saw after the training, only in the neurofeedback group, an increase and an effect on behavior. Well, and this is a consensus paper on the reporting and experimental design on clinical and cognitive behavioral neurofeedback studies. So this is a joint force of very different researchers across the whole globe, so to say. I wanted to figure out the exact number of researchers that participated there. On the bottom, you can see that there were a lot of modalities represented. So this is also something that I really enjoy, to speak and interchange with not only people that do EEG and neurofeedback, but you see here in orange, there are the people doing fMRI, also NIRS and multimodal, even multimodal neurofeedback was integrated as well, with different research domains. So the majority were researchers in the clinical domain, and we had really a lot of countries. So this is a joint force, and what we wanted to do as researchers all together is to encourage a robust experimental design and to also have a clear reporting. And with this, we would like also to advance the whole neurofeedback field. So in future studies of neurofeedback, we want to take care really about the methodology, so that we, in the end, also can see, okay, what contributes to the benefits of neurofeedback. So here on the left side, you see a little bit what mechanisms there might be, but I thought I would like to share this with you, because I guess that you will see in future also some other of our neurofeedback colleagues here.
Yeah, brilliant. Thanks, Stefanie. It's so important that we know that all the researchers like yourself have some sort of consensus and consistency with your methodology. You're following the same sort of ethical standards and design standards, so that we can sort of compare and keep the scientific integrity there. So that's brilliant. And so what's the focus of your current research at the moment, Stefanie?
Well, I'm focusing content-wise on executive functions, as you noticed. So I think this is one of the most exciting cognitive functions in my view. And you might maybe ask, okay, why is it the case? Is it good for solving puzzles? Does it have any implication? And this is exactly what I like. So executive functions enable you to follow on your goal-oriented behavior. So it has an implication for everyday life. So again, here, so the differences in executive functions will lead that maybe one person is, it's for him easier in an academic domain. We're also in the moment investigating with the collaboration partners pilots and their flying behavior. So we want to see, okay, what is the association of executive functions and flying? Are there better pilots than pilots that have a little bit more difficulties in executive functions? So executive functions are very important, and also when it comes to the clinical domain. So I know that you're interested also in the prognostic value, but for instance, executive function, if you would take them and measure them at the beginning, it can give you a direction of how well patients also are successful in their therapies in the medical domain, but also in the more cognitive behavioral therapy domain. So this is quite interesting. And I think, therefore, it has such a relevance for everyday life, for the functionability in the social domain, in the emotional domain, that for me, this means, yeah, I want to spend my research time on this topic. All right, so here I have a slide for you, and I really like the graphic. So what you see here is a music conductor orchestrating an orchestra of the brain. And this is a very nice metaphor of executive functions. So sometimes you hear also that executive functions is like a conductor, and responsible and enabling, for instance, let's say memory or attention. And here I have I just put some definitions. So to sum it up, so executive functions is a collection of interrelated higher cognitive function that control primary processes. And because of this, we have the metaphor, I think, of a conductor really fits. I told you that I really like executive functions because it's association to everyday life. So I listed some where we know that there is a relation. So it's academic performance, it's occupational functioning, social functioning, and so on. So you see that it's also relevant for healthy participants. So we have, for instance, we did a study regarding executive functions and the association to academic performance in students, in a student population. And I think also when it comes to neurofeedback for the improvement of executive functions, is of course important for patient groups, but it is also relevant for healthy participants when it comes to peak performance training. So for instance, in pilots and young pilots, to then in the end have the goal to have a safe flying piloting for them. Yeah, and in general, I do research in 2 domains. So you might have noticed that already a bit, so that there are 2 big domains. On the one hand, we are very interested in the neuroscientific research on executive functions. We want to know how the brain implements it and increase the understanding. But on the other hand, what is important is, okay, how can we use it for application in the clinical domain, also for peak performance training. So for us, also translational research is very important. And here one big domain is the development of brain trainings and how to assess them also for therapy and mainly neurofeedback. Currently, we have a very big study that we're finishing, and this is also again a frontal midline theta training, but in the clinical domain. So we invite people that reported on executive function problems. So here you can also see on what they report, where they would have difficulties. And a lot of people come that want to take part in this neurofeedback experiment because they have the feeling they have difficulties in planning and sustained attention and time management, emotion regulation, working memory, and also simply to start a task. So we screened them with the brief A, and then they were invited to take part again in an 8-session neurofeedback. We assessed, as you have seen before, the 4 different tasks on executive functions, because they represent very nicely the most important subcomponents of executive functions. This is currently something we're finishing. So if you're interested, we will send you then also the article about it. And I think the part here is one of my favorite graphics. And what you can see here is I'm moving a little bit back and forth, and I come to patient groups. But with this slide, you also can see why neurofeedback and why the improvement of executive functions is also good for everyone. So, and here, what I like about it is that you can see that there is a kind. So one on one axis represents how brain functions, how networks can be affected. and so that they don't work well. And let's say this end could be someone, a patient with a lesion. So from this, you would not expect a very good cognitive performance. And on the left here in the green corner, you can see the optimal performance of a healthy young participant, let's say. And then what is interesting is that if there's a stress, a lot of emotions, task overload, fatigue, or also environmental aspects, this will gonna stress also healthy participants out to the way that the executive function performance will decrease and will move along on a dimension into the direction of clinical population. So therefore, I think this reflects again how important neurofeedback is also for healthy participants. Yeah, so these are some examples what we do. Also regarding the neuroscientific research on executive functions, currently we assess also how the brain communicates, so how the brain areas communicate. So this is a little bit about connectivity. And this might be a next target also for neurofeedback.
Excellent. So, so this is what you've been doing at the moment. That's a great overview of all your previous research. You've mentioned executive function. You've mentioned performance. Are you going to stay in that sort of domain moving forward? What sort of future aspirations do you have with your research, and will it expand outside executive function, or you'll try to remain in that area?
Yeah, this is a really, again, one of the most difficult questions, I would say, but definitely I think there are two ways of answering it. So I'm wondering, what are the conditions? Also, how do I want to perform research? And this is kind of the meta level. I think for the future context in which I would like to work is, I would like to contribute also to and strengthen the research communities. For instance, we have currently nice joint collaborations that are there are many different disciplines taking part. I think this is really strengthening communities. I think as researchers, we create a lot of knowledge, but what is important also is that we try also to offer solutions, and that we would like to bridge also the research practice gap. So very often research or taking it over to application takes a lot of years, sometimes even decades. And I think this has to stop. So I would like to bridge this gap. And then of course, content level. Yeah, there are definitely, I would like to continue with executive functions with neurofeedback. Would like to follow on with understanding the training and the response, and why some people are better, why others respond less to neurofeedback or to any training. And of course, also to identify predictors, because this will show us how and where we can assign then participants to a protocol that works better for them. And this is also the next point. So we would like to develop further protocols, maybe about connectivity. This is one possibility. And of course, it's for us important also to really develop good and efficient neurofeedback protocols that are effective in everyday life, that can be translated, that can be also used outside the lab, in real life and in the clinical domain, but also maybe as a training.
And so that's what you're going to be focused on personally. What about the field in general of neurofeedback? How do you think things are going to move over the next 10 years? And how quickly do you think these things are going to move into clinical practice? Do you think technology has a role to play there as well?
Yeah, so I think the last years have been very, very exciting. I have also a slide that I would like to show. All right, so here you see a graphic, and in this graphic you see that there is this really strong increase over the last 3 decades on papers on neurofeedback. And the colors represent also everything what is the method. So you see that there is a lot about fMRI, not only EEG. So this is a definite increase. There is also non-EEG neurofeedback like fMRI, but also like fNIRS. And what we have seen also the past, I don't know, let's say 5 to 8 years, is also that there are a lot of advancement in the method. So suddenly there are articles about connectivity, implicit neurofeedback without telling someone that they have to do something, decoded neurofeedback, so detecting specific neurostates, but also MRI-informed EEG neurofeedback. So there is a lot of advances in the methods. There are more methodologies than only EEG. And of course, there's also innovation, big steps in the technologies. So there are new EEG systems that are very small, that are portable, wireless, that makes it easy, for instance, to be used at home, but maybe also to be used with kids more easily. And so it's very exciting, I think. And I brought you one of our clips, and I'm not sure if you have seen it, Trevor, but on our homepage we have a clip, and it's called Science or Science Fiction. And it shows a little bit, there are a lot of commercials outside about neurofeedback and what you can do, and that you apparently can switch off your lights in your house, and that you can use it for smart living. And this is, if you're interested, just take a look there. It's the question about science or science fiction, what will be possible, what not. And it's a little bit going into your direction. But I guess in the next 10 years, we will see more also probably transferred to the clinical application. I put also a nice last article where you also contributed, and I like it for several reasons. So I think what you see here is it's neurofeedback as applied in ADHD. And I think here this article mostly shows the strengths of personalized neurofeedback applications, and also multimodal. And I think this is also something that shows a direction I would expect that this is in the next years implemented much more on purpose.
Great. Yeah, thanks, Stefanie. It was a pleasure to work with you on that review. I hope we have lots more involvement with you in the future here at Neurocare, because you're doing a lot of great research, and obviously have an eye towards the clinical application of it, personalized medicine. So really, we have a lot of common goals moving forward. And it's been a pleasure to talk to you today. Thank you very much for your time, and I hope we get an update from all your research in the future.
Yeah, thank you very much for having me here, and I'm very glad to come back and tell you more. Have a nice day.
Fantastic. Thanks, Stefanie. See ya.